All righty. Now we're recording. So thanks again, everyone, for joining us. And just to review for those who may be new to Teams, the toolbar is pictured here on how to access all the different pieces. And for those that are using the Teams app, the toolbar is in the top right. And if you're joining by browser, it should be at the center bottom of your screen. You may have to move your mouse to make it appear. Just make sure that your microphone is muted if you're not speaking. And then if you have to ask a question, you're welcome to just chime in, unmute yourself and chime in that way when we get to questions. And then you can also use the chat or raise hand feature to join the discussion as well. Each presenter today has about 15 minutes of time with 5 minutes for questions, and we're full up till 3 p.m. today with presenters. So lots of information to cover today. So our agenda here, we'll have Dan with Sherburne County SWCD talking about his infographic for early detection and response for Sherburne County. We'll have Josh Stumke with University of Minnesota Duluth talking about using eDNA to detect AIS. And then we'll have Megan Weber with University of Minnesota Extension talking about Starry Trek and AIS detectors. And Meg Durr will jump in real quick just for 5 minutes to talk about the Stop Spiny campaign and Spiny water flea volunteer surveillance pilot that will be happening this year. And we'll take a quick stretch break and come back with some organisms in trade topics with Chelsea Blanke with the DNR and Tony Ruff with Hennepin County. So got a full palette of stuff to talk about here today. And with that, I will kick things off and over to Dan. So stop sharing my screen, Dan, and you can go ahead and get started.
Sure. Good afternoon, everyone. Dan Spolka, Senior Water Resource Specialist with Sherburne Soil and Water Conservation District. And that job title includes overseeing our aquatic invasive species prevention program. The project I'm going to share with you today is a fun little project that the Soil and Water District took on in coordination with our Sherburne County Coalition of Lake Associations group. So to set the scene for Sherburne County, we've got 127 lakes. We've got about 16 of those lakes have public access, and then there's 10, I believe, active lake groups. And they formed a coalition of lake associations a while back, a couple of years back, 2017. And have been looking to engage in projects that benefit all of the lake associations in the county as a whole. We got on the topic of aquatic invasive species and early detection rapid response. So the county itself has a early detection rapid response plan. It's about 40 pages long, a lot of text, and it's a great document, if I do say so myself. I think what the lake groups were looking for was something that would be unique to them, something that would be a little more user-friendly for non-industry professionals, and something that had a bit of a fun visual element to it. And so the COLA and Soil and Water staff, we started discussing how could we make that vision into something that would be usable for everybody. So the idea was to come up with a template document that could be modified to each local group's individual situation, given their budget, their level of participation with the organization, etc. So what I'll share with you today is a draft of that project. And let's bring that up. So you should be viewing now, this is the Sherburne County Coalition of Lake Associations website. There is a person involved in the group, Cindy Plant, who has a graphic design background. So she has been putting her work, her professional efforts to use on all of the publications. So you can find this site yourself. It's sherburnecola.org. And actually, if you have any comments or feedback on this project or other things, I'd be more than happy to have a phone call or email sent my way. Like I said, it is in draft form. Tina and a few other DNR staff did provide some initial feedback, so we'll be incorporating that soon. Under the About tab, we'll navigate to projects. And here we are. We've got a number of different projects outlined. This first one here is our early detection rapid response activity. And again, we wanted something visual, something that's not only user-friendly, but could be something that would include additional layers of complexity for those that want to dig into it. And I'll discuss that as we go along a little bit further here. But at first, we have two links that you could click on. So we have print-friendly handouts, and this just connects to a Google Drive. And basically, the bulk of this effort contains 3 different handouts. Again, a visually aesthetic handout that's a little simplified for folks that are just getting their feet wet, so to speak, with aquatic invasive species. But it responds what we call a first responder or a lake user to the initial stages of AIS identification, detection, and reporting. So very clear steps on collecting a specimen, how do you record it, and who do you report it to? And I'll go through these initial slides a little quick, and then we'll go into something else. Following the first responder, there is a evaluation team, and this gives some very brief details on what would an evaluation team include. So it includes perhaps a lake association member, a RGU, that's a responsible governmental unit, and then AIS staff. The responsible governmental unit could include watershed districts, it could include county staff, soil and water staff, etc. But then we go through the steps of how do you understand this infestation further, and how do you, once you've reported it and identified that it is in fact a non-native plant or animal, again, what are the steps that need to be completed? And it's outlined here in a bit of a checkbox format. And then following that, we have the action team. The action team may consist of the same individuals or also some additional folks such as a contractor. But at this point in time, you would be creating maps, communicating with others, evaluating your options for how you would manage the infestation and what direction you would go, and developing a long-term strategy. So again, this is something that is pretty simple at first glance. So the average lake user or lake visitor could pick this up pretty easily. These are formatted so you could print them out and include in your tackle box or in your car. And so there's benefits to having a printed version is something that we thought of. If you click on this interactive universal action plan, so now we have a PowerPoint slide deck. You'll see those same cards again, but this allows somebody that has a bit of experience or a bit of interest to dive a little bit deeper into this process to go further. So again, you have this first responder tab, which identifies what you should do if you have a suspect animal or plant in a lake, collect a specimen, record it. But maybe you're looking for a little bit more detail in terms of how do I identify this thing that I've found. You can click on this link and it will take you to a useful web page, such as the Department of Natural Resources Guide to Aquatic Invasive Species. Here you can scroll through and get some information as far as how to, helping you to identify what you may have. There's other links, including reporting. So if you are going to report to a lake association, maybe you don't know necessarily who your lake association contacts are. Click here, and you've got the list of all of the member associations within the COLA. On this second page, again, this is the evaluation team. So you've got your first glance checklist of different things that you would need to do as far as a standard AIS early detection and response scenario. But in assembling your AIS evaluation team, there's a link here that will bring you to a spreadsheet where if you are the Ann Lake Improvement Association, you can fill out this information to have all of these people's contact information ready to go. So in here, you'll see who is our area AIS specialist. That's Christine Jurek. So here's her phone number, email. Who is the county AIS coordinator? That's Dan. Again, phone number, email. Additionally, if you've got somebody on your lake association that is the finance specialist, the finance person of your operation, insert their contact information there. It's ready to go for you. There's also a link here to EDD maps, which is helpful if you or somebody on your team is looking to report something that way. And again, same information, but the opportunity to dig a little bit deeper for those that need to. On this third page here, there's some helpful links as well. So this is the action team. This is the group that's going to be, again, digging a little bit deeper into the extent of the problem and how do we develop a strategy to address what we have. So creating a map, how do I do that? Here's a great link created by Tina and others at the DNR for guidance for creating a map for assessing the situation and developing a map to get a understanding of what's happening out on your lake. There's additional links on here for permits. So we've got a link here to the MPARS website. And overall, the goal again with this project is to create something that gives all potential users of a lake an understanding of what happens when you find a suspect zebra mussel or a suspect starry stonewort. It outlines very clearly and in a creative and fun way, hopefully, what are the steps that I need to go through? And for somebody that is involved at the Lake Association, as far as decision making, they have the resources that they need available to them very quickly to understand who is it that I contact, where are some resources as far as developing a map, and somewhere in here, let's see, maybe it's on this page. We do have a list of, here we go, reference local case studies. So as a part of this effort, we did reach out to some neighboring lake associations. Wright County folks were very helpful, and a few others, Hennepin, Stearns nearby. So we assembled some case studies to get an idea of what are some potential approaches and costs. And so we've got a few different early detection responses. So we did send out a questionnaire to a number of people to ask questions such as, have you found something? How was it found? Have you treated it? What were the associated costs on a per acre basis? What were the options at your disposal? So it's a spreadsheet that outlines each of these early detection situations and tries to give a picture of what were the options available, what were the associated costs. Again, this is something that's hopefully providing real world cost estimates to the folks that would be considering those decisions should they find something along the lines of an AIS in their lake. So it was a fun project. It has gotten pretty good response from our lake associations. There, again, with so many different groups and developing a template for each of them that they could use. There was some folks that are pretty tech savvy, wanted something with clickable links and spreadsheets, and some folks that wanted something that they could really just have in their tackle box when they're out in the lake and ready to go should they find something. There was another group that wanted to post that first user, that first responded slide up at the town hall bulletin board. So the hope was that this was something that could be usable for the average lake user and somebody that again is intimately involved with decision making and permitting and financing management of AIS. So if you get a chance, log on to the website, scroll through, and I'd love some feedback, good and bad and in between. So any questions?
Just waiting to see if I don't see any questions in the chat. Oh, Jacob asks, where is this available on the web? And if you scroll up in the chat again, I can copy paste it into the chat again here for you. There we go.
Yeah, great question. The Sherburne Cola website, sherburncola.org. And I can either, Tina, if you could throw that in the chat, or I can once I stop sharing my screen. And then under the About tab and Projects. And like I said, it is a work in progress. We do have some comments from Tina that we'll be integrating here soon. But if you see other things and have questions or comments, feel free to shoot those my way.
Awesome. Well, thank you, Dan. That was as we all know, early detection and response is quite a complex thing, and that every situation is unique depending on the circumstances. So being able to boil that down into a few check boxes is pretty significant in itself. And then providing those additional resources for those who really want to dive in deeper. So it'll be interesting to hear how it, hopefully you don't have to use it at all this season, right? But if you do, if you do, it'll be interesting to hear, how it worked for everyone involved, and if, any feedback that you get there, and maybe we can learn more if and when it needs to be used.
Yeah, thanks, Tina.
All right. Well, then I think we can jump on to our next speaker here. So thanks again, Dan. And Josh, if you're ready and there to start sharing your screen and starting your presentation, feel free to do so. And also make sure to introduce yourself as well.
All right, thank you. Okay, has my PowerPoint showed up yet?
Not yet.
Not yet. Oh, there we go. All right. I was sharing the wrong thing. Learning how to get through between Zoom and Teams can be challenging sometimes.
It sure can. And then WebEx and yeah, all the things. They're all put in different places. So we're seeing your full PowerPoint application with the slides on the side.
Oh, with the slides on the side, huh?
Mm-hmm.
Oh boy. Well, I don't know how to get through that because for me, it looks like full screen.
Yeah, you might just be sharing the wrong screen.
Josh, this is Doug. I think you can just hit view.
And oh, you think so?
Okay. I think so. Yep. Hopefully that does it. You might have to go back out of it and come back in.
Oh, great.
Give that a shot. I've had the same difficulties.
We can read it pretty well if you can scrunch the slides off to the side, and I can see the content pretty okay. And there's someone in the chat saying Alt Tab will cycle it. Hold Alt and hit Tab. Nothing's changing. That's
I
okay.
know. I looked at it. It was still just showing the PowerPoint application, but when I change
Yep.
my view of it, it doesn't seem to change it the way you see it. Oh well. Well,
Yeah.
we'll move on. Challenges with technology. So I will, I guess, talk about a project that we've been working on for the last year called Optimizing eDNA Monitoring for Multiple AIS. This is 1 year progress update from a team of collaborators, including myself, Josh Dumke. I'm a senior research scientist at the Natural Resources Research Institute, and I'm the PI on the project and leading the team out of NRI. And our co-PI, Gretchen Hansen, down in the Twin Cities. Chanlan Chun is the microbiologist working out here at NRI, overseeing all of the lab processing. Eric Larson is a invasive species specialist using eDNA to detect invasive species out of University of Illinois, and we're actually using primers that he developed for rusty crayfish. Anna Toch is a grad student working at NRI. Chris Rounds is a grad student out of the Twin Cities campus. And Adele and Jenna are lab and field technicians out of NRI and Twin Cities teams, respectively. This project was funded by the Minnesota Environment and Natural Resources Trust Fund as recommended by the Minnesota Aquatic Invasive Species Research Center and the Legislative Citizen Commission on Minnesota Resources. So the background of the problem is that aquatic invasive species continue to spread among Minnesota water bodies. And we all know this when we get the DNR bulletins throughout the summer of where starry stonewort or zebra mussel have been found in new lakes. But we also don't get those updates for every species. Certain species flag the infested waters designation and some do not. So there's species that we see that are being moved around the state, and there's that like we aren't getting a lot of information about.
Hey,
And
Josh,
invasive species early detection is critical. Yep.
If you're moving your slides, we're still seeing the first one.
Oh, really?
Yeah,
Okay.
I think you have to select them in the PowerPoint application itself. Thanks.
Huh.
Yep, now it's on background of the problem.
Really? Okay.
Mm-hmm.
All right. So the I guess the point is that early detection using traditional methods has high cost and limited scope. You can only go to a certain amount of lakes each year, and usually the traditional methods are specific to one species at a time. And environmental DNA or eDNA might be the answer. eDNA is DNA released by organisms into their environment. So things like skin cells, mucus, and feces, anything that is left behind by an organism. So how might eDNA help? Collecting water is an easy way to get eDNA. And you can look for several invasive species from each water sample. But eDNA methods are not standardized, which introduces uncertainty about how well eDNA performs. If lots of different people are using different methods, they might understandably be getting different results about making detections. eDNA methods need to be optimized for multiple invasive species before broad application. So that's the point of our project. We want to optimize the field and lab methods to maximize detection probability of multiple invasive species using eDNA. And we're going to determine this through two complementary activities. Activity one is led by Gretchen Hansen and her grad student Chris Rounds down in the Twin Cities. How does lake character and seasonality affect detection probabilities of multiple AIS using eDNA? And lake character are things like lake size, maximum depth, their trophic status, and things like that. And seasonality is between ice out in the spring and ice up in the late fall, when during the open water period should you take samples, and does it matter? Activity two is being led by myself and the NRI team. What is the optimal combination of eDNA field and lab methods? So there's a lot of choices to make when using eDNA as a method. How much water to collect, how to filter that water, how to do the extractions and the quantification. All of those have multiple choices. and we're testing several of them to see which ones offer the best opportunity to collect DNA from the field, called harvest, and the best way to quantify it. So if you have a limited amount of DNA harvested from the field, making sure that you have sensitive enough methods to detect that DNA. Activity 1 methods are spread out among the lake, where we have 20 lakes selected. 13 were sampled last year, and 8 more will be sampled this summer. And you can see that 13 and 8 don't add up to 20. Well, that's because last year we sampled Lake Owasso, and in October, a DNR bulletin came out saying that zebra mussel were found in Lake Owasso, and that was right at the end of our sampling season. So we decided to go back to Owasso this summer, so we have 2 years of data. And the lakes selected vary in size, their trophic status, and region of the state. And we picked these lakes which offer a mix of known presence or absence for our 4 target invasive species. And I should say known absence is kind of a misnomer, at least not detected at the time that we set up this project. As I mentioned, Owasso, before we had started sampling it, wasn't known to have zebra mussel. It may have had zebra mussel that whole time. So we're looking at lakes that have at least known presence and reasonable expectation of an absence for common carp, zebra mussel, rusty crayfish, and spiny water flea. Each lake is visited 5 times from April through October to represent stratified and non-stratified conditions. And at each lake visit, we collect 250 milliliter water sample grabs from the same 10 locations. 5 of the locations are near shore and 5 are offshore. We collect covariate water chemistry data like pH and temperature because those can influence how long DNA persists in the water before degrading, and major cations and anions because they can inhibit PCR, which limits your ability to quantify data, quantify DNA, even if you had captured it. And all water grabs are treated the same. They're filtered through a 1 micron cellulose nitrate filter and extracted with a Kaigen Power Soil Pro kit, then single species qPCR to quantify target AIS genes. So we do the extraction from each sample, and then we run qPCR 4 times, once for each species. So thus far, we've collected 975 samples from 13 lakes. 650 of those are for eDNA, plus field blanks and samples for cations and anions. We've had the cation anion samples done for quite some time. The eDNA lab work continues for 2021 samples. We're over halfway done with the extractions, and qPCR has been started and is underway for common carp and rusty crayfish. And the lab has really been running at full speed for about 5 or 6 weeks. So this process of getting through last year's samples is just really ramping up right now. So presently, I don't have enough results to discuss how lake type or time of year affect eDNA as a method. We're in the middle of getting all of these samples processed, and there just wasn't a complete set of either one whole species or one whole lake throughout a season to talk about activity 1. But honestly, that's going to change in just a couple of months, and then we'll have something more that we can update about. For activity 2, we sampled 5 lakes in 2021. These lakes, again, were selected which offer a mix of known presence or absence of our target invasive species, but as well as a range of their abundance. And an example of that is the difference between Lake of the Woods and the St. Louis River for zebra mussel. Lake of the Woods, veligers have been detected, but adults still have been eluding searches. In the St. Louis River, there's zebra mussels everywhere. And in fact, some of our zooplankton tows, we had almost 40,000 veligers in a single zooplankton tow. So there's a very high abundance of zebra mussels in the St. Louis River and a relatively low abundance in Lake of the Woods. We wanted to capture that. And this activity differs a little from activity 1. Each lake is only visited once, so we collect a lot of samples at one point in time. And sample locations are paired with traditional invasive species surveys. Many paired test groups of DNA harvest. So that's the different ways of collecting the samples. Different volumes of water and different filter types are being paired together to see which ones are performing better. And we have DNA extraction and quantification test groups. So more of the lab methods. If you choose different ways to do extractions or different methods of quantification, do you find that one of them performs better than the other? So a quick synopsis of our field sampling looks like this. Near shore, surface water grabs, offshore surface water grabs, and offshore benthic grabs, where we would lower a Van Dorn down and collect water samples near the lake bottom. Test group A is the same at all 3 points. A 250 milliliter grab filtered with a 1 micron filter and preserved in ethanol. And that's the same that was used in activity 1. Test groups B, C, D, and E are all different variations of collecting samples. Collecting 1 liter of water, or using a 5 micron filter, or using silica beads as a desiccant, or preserving the sample in CTAB rather than ethanol. And CTAB is a buffer that can, over time, will cause cells to lyse and release more DNA into a sample. So I'm not going to talk about the sample groups B, C, D, and E today because we don't have enough of those data processed yet. And at each sample point, we did traditional AIS monitoring. Near shore, we did horizontal zooplankton tows, snorkeling or shoreline searches for adult zebra mussels or rusty crayfish, and we set crayfish traps. Offshore, we did zooplankton tows again, as well, horizontal as well as vertical. We used an underwater camera, which is that white box I'm holding up in the bottom right corner, and ponar grabs if the camera wasn't used. For preliminary results, I can talk about what we found with rusty crayfish from 2 lakes, the St. Louis River and Lake of the Woods. That's the extent of the samples that we've completed thus far. And just for test group A, so all of the same water volumes, the same filter, same preservation, but just at different places. So near shore surface water, offshore surface water, and benthic water. In Lake of the Woods, we had points that were spread out as near shore and offshore. Near shore points are 1 through 5, and offshore are 6 through 10. When we did our physical surveys, we didn't actually capture any crayfish in traps that were rusties. We had virile crayfish, but no rusty crayfish. And when we did the underwater camera at point 8, I had seen a crayfish, but I couldn't actually verify if it was a rusty or not. At point 7, which is the green triangle, I actually did chase down a crayfish and get a view of its side and could verify it was a rusty crayfish. So we had 1 positive sighting of a rusty crayfish using physical detection methods. When we bring in the eDNA sample data, we see a lot more is added to the map. There were 6 positive detections using eDNA. The positive detections from benthic samples, or those collected with the Van Dorn near the lake bottom, are the purple circles, and the red circles are surface water. So you can see some of the offshore sampling points are getting positive returns with the samples collected with a Van Dorn near the lake bottom. The nearshore points tend to be having positive DNA hits off surface water. And the 0.6 is the one where surface water and benthic water both had positive returns. And that point was one of the shallowest of our offshore points. It was about 19 or 20 feet deep. And for about 2 days before our visit, we had extremely high winds, about 25 mile an hour, and there were about 4-foot waves in that part of the basin. So perhaps that part of the basin was mixing a lot more than some of the other offshore points. In the St. Louis River, we didn't catch any rusty crayfish. Our crayfish traps caught gobies, and crayfish were present because I had captured by hand a northern clearwater crayfish, and we had seen other crayfish scooting around to escape us, but we didn't actually capture any. And we couldn't use the underwater camera effectively here because the water was too turbid and the current was pushing the camera around. So we were relying more on ponar grabs, which are not going to catch a crayfish. Using the eDNA as a detection method, we had, again, 6 positive returns for rusty crayfish DNA. In the upper river, we had 0.7, had a positive hit on a Van Dorn. All the other sample points had positive returns for surface water. Now, that down in the lower harbor at points 8 and 6, those had surface water and benthic water sample grabs, but we didn't get positive returns on the benthic water grabs. Now, there's only speculation about why that might be at this point, but we do know that the lower harbor has perhaps more mixing with Lake Superior. And as a system, as a riverine system, that might influence where some of these DNA positive returns come from. So, in summary of our preliminary rusty crayfish data, eDNA was more sensitive than traditional physical monitoring, and where we collect samples for eDNA could affect detection. In Lake of the Woods, for example, there were more rusty crayfish eDNA detections when samples were collected near the lake bottom. And this was just a subset of samples which may not even be the best methods. Other test groups may perform better, like 1 liter water grabs, or doing the quantification with droplet digital PCR, which is a much more sensitive method with lower limits of quantification than a single species qPCR like we processed so far. And still to do, we have 8 more lakes for activity 1 that we'll sample this summer, and hundreds of samples still to process in the lab. But the answers are coming. And by the project end, we expect to know which lake types will be more suited for eDNA as an AIS detection method, when and where in a lake to collect water samples for eDNA, which of our tested field methods work best for capturing eDNA, and which lab methods are most sensitive at recovering and quantifying DNA. Our goal is to create method recommendations for the field application of eDNA when monitoring AIS in Minnesota, so that managers, specialists, and NGOs have guidance and confidence when using eDNA as a monitoring strategy. An eDNA best practices guide, which will describe our findings and recommendations in plain language, is one of our other outcomes, in addition to our expected publications, student thesis, and presentations to groups like this. So with that, I have just a couple of acknowledgements for some of our field team members who helped us in collecting samples in the lab. Minnesota Sea Grant offered the underwater camera for me to use to view lake bottoms, and Ramsey County was working with the Twin Cities team to collect eDNA samples from a subset of metro area lakes. So their help to collect samples was greatly appreciated. So thank you. And if there are some questions, I can entertain them. And I have no idea what everyone can see right now. So just tell me if I need to move around on different slides.
Yeah, you're on the thank you page right now. And it looks like, Doug, you have your hand up. Go ahead.
Yeah, thank you, Tina. Yeah, nice job, Josh. Yeah, you're right. For those that may not know, the St. Louis River and Lake Superior oftentimes will get a wind-driven seiche, which will drive water from Lake Superior, and because it's colder, will mix, especially in the lower harbor. But it'll also go all the way up to Spirit Valley Marina, which is just well where the big island is, up into that area. So it'll really affect, I think, the impacts on the sensitivity of picking up the eDNA. Josh, I was also wondering, are you collecting substrate or characterizing the substrate in the general area where those samples are taken? We know that rusty crayfish prefer cobble area where they like to hide out in the interstices between the rocks and that sort of thing, as opposed to maybe the mud flat areas.
Yeah, those appear as notes in all of my sample points. In a lot of cases, when I would put the underwater camera down, the offshore points are all almost, it's like pudding. I mean, it's the silt is so loose that you could tell that just driving around, there's nothing for the rusty crayfish to really attach to. And
Right.
you almost can't even tell when you bump bottom with that underwater camera because it's just like flocculent, fluffy stuff. So yes, that does get characterized as notes. Whether we're near shore or offshore, we either see the bottom when we use the underwater camera, or we pull up really, really fine, pudding consistency material with the ponar.
Sure. And rusty crayfish generally don't burrow, but they are known to sometimes burrow.
And I had seen in Lake of the Woods when I was chasing down some crayfish that burbot appeared to have made some holes in the bottom, and they would kind of lay in this trough that they had made. And we had seen some crayfish duck into those, and I used them as hiding spaces.
Interesting. Thanks.
Any more questions for Josh? All right. Well, thank you again, Josh. We're excited to see the results of your project, and I'm also really interested that you're making kind of a plain language field guide and lab guide to come out of this as part of one of the deliverables. That's really exciting to hear as well.
Thank you.
All right, Megan, you ready to go?
I think so. Let's see if I can still do the share. Hopefully I don't have as many issues. I think it's sharing. Is it sharing the right screen?
It's showing just your first title screen.
Hey, all right.
Yay, technology.
Right. So, yeah, Megan Weber, I'm an extension educator that focuses on aquatic invasive species issues. And yeah, I came to chat about a couple of our programs. I first also wanted to acknowledge the rest of the team. So the primary people that I work on these programs with are Dan Larkin and Pat Mulcahy. And we have kind of this suite of programs, and I think many of you are probably familiar with, at the very least one of these. I see lots of familiar names in the chat, but I'm really going to focus on just these first two, our detectors core course in the volunteer program and Starry Trek. And I'll start with the core course. So this is the program that started it all, I guess, with our set of programs. We started this, the first official launch was in 2017 in the spring. And our goal was to really increase the volunteer capacity to help work on aquatic invasive species issues in the state. So we created a course that kind of follows a Minnesota Master Naturalist or a Master Gardener style, training and volunteer, I don't know, organization maybe. Where we use this flipped classroom model, which, I mean, the pandemic brought all kinds of changes into that too, but it has this online portion that's done at a self-paced style. So you just kind of go through, work through the content. We cover everything from kind of the basics and introductions to basic aquatic ecology, basics about aquatic invasive species and impacts, and then start to get into a lot more specifics about the aquatic invasive species we're really interested in you learning about, how to identify, how to discern them from some very common native lookalikes, how to report a potential invasive species and to make a good report, regulations in the state, how to do monitoring, things like that. And then we bring everyone in for a workshop, and now there's both virtual and in-person options for that. And in that workshop, we really get into some literally hands-on in the in-person workshop, and then as much of a hands-on as we can make for the virtual one, for some time with kind of that group or cohort of people to learn through it. And ultimately, the goal then is to have this group of volunteers that has this really good base level of knowledge to go out and volunteer in the ways that mean the most to them. We generally will kind of classify those into groups like stewardship, which is kind of hands-on, digging in the dirt type work, program support, which we would consider any of the things that make an aquatic invasive species program go. One of the pictures here is volunteers who came to help me stuff bags for Starry Trek. So it might not feel necessarily like AIS work because it's in an office. This was in the basement of where I work, but really critical things that are needed for these programs to run. Citizen science, which Starry Trek itself would probably be, or helping researchers out in the field, and education and outreach. So where we've gone, and in a couple of slides here, I've added coronavirus asterisks on some of the data, but we had a lot of really great growth before the pandemic hit in terms of the number of hours that we were seeing. So 2017, I'd mentioned, was the first year. And then through 2019 was kind of like all the seasons that we got that did not have a pandemic impacting them. So we started to see this really great effort. The pandemic hindered a lot of that because lots of hours related in particular to education outreach, and lots of those programs have kind of been gone in the sense, in like the way that this group was able to engage with. And so, you know, lots of things were gone. I'm still super impressed that there were over 2,000 hours that people found things to do, that logged into 2,000 hours during kind of the 2 pandemic years I have marked there. I'm super hopeful that maybe this year I can have a bar that doesn't have a little coronavirus next to it. But we take that and use this number that is created by independent sector that kind of puts a value on just this is just the time. So we're not talking about ecological services or anything like that. This is just the value of the time that people are putting in. And it's to date now, as of February of this year, was over $600,000 worth of time that people have invested to help kind of bring this extra capacity to aquatic invasive species work in the state through this program. And I put, like, there's a little QR code and the link underneath it. If you want to get updates about this program, you can scan that and enter your email, and we'll send out notices when there's registration. And by the way, it's open right now. So and we actually added a track. Last year was the first year that we added a track in for professionals. So if you're not a volunteer, but like the content still sounds interesting, so we took a spot where we kind of switched out some content to make it a little bit more relevant to like a non-volunteer. So we have 2 in-person workshops this year in New Brighton and in Alexandria. And then we have 2 virtual workshops. And those are, you know, anywhere you want to be is where they take place. We'll ship you a box of goodies to use during the workshop. And those are both towards the end of May. The other couple of things that I wanted to like quickly say, I'm looking at the time, to quickly say about this program, in addition to what I have already, I guess, is we do have a couple of other ways to engage. So one thing that we noticed is that we started to see a lot of sponsorships come through, where like an organization was sponsoring people to come, and we started to get a lot of requests to like, can you bring the program to this location? Can you bring the program to this location? So we tried to find a way to be able to accommodate a lot of that. The sponsorships are still easy if it's just a couple of people, but we created what we call group workshops. And really what that is instead of having to like sponsor individual people and send them to the places that we've picked, you basically just buy out a workshop. And I think when we did the math on it, and it breaks even at about like 15, somewhere between 15 and 17 people typically, where if you think you're going to sponsor that number of people or more, then you can have up to 25 people in the workshops, and that group workshop might be a nice option for you. Also nice because you get a lot more say on where and when that workshop happens. So there's that. But the other piece that I really want to say, especially as I mentioned, you know, we've had a couple of years with fewer volunteer hours reported, is this volunteer network is really open to the state. So anybody who has activities going on that could use some volunteer support, if you connect with me or Pat or Dan, we'll advertise that volunteer opportunity to that volunteer network to help get them engaged near you. And you know, we have volunteers across the state. I think that in general, people are starting to get kind of ready to go back out and do things like this. So I'm really hoping that people around the state will recognize that and maybe see an opportunity here, and also have another way to kind of start re-engaging a really dedicated and wonderful group of people. I think that's all I have for this one. So I'm going to move on to Starry Trek. It looks like I put slides out of order. I don't. Anyways, oh no, maybe I didn't. Okay, anyway, sorry, Strek. I think if you were on an earlier one of these, where Dan Cebulka and I were talking about Curbicula. You probably heard like a really brief intro to Starry Trek. But this is a one-day event. This is a statewide search for starry stonewort and other aquatic invasive species. And we were kind of inspired to create this after kind of a flurry of starry stonewort discoveries in 2016, where suddenly we jumped from like one lake in the state that we knew had it to, I think it was 6. I can't remember now. But it was a pretty significant jump, and we really thought, it's probably in some other lakes, and people just don't know what they're looking at or looking for, or don't realize it, or we just haven't looked. And had just created this volunteer program and realized that this might be a really great way to kind of mobilize that group of volunteers. And it turns out that we've been able to not only mobilize that group, but lots of other people who are really interested in maybe like just kind of this one-day thing versus a long-term volunteering effort. So the group in the photo here is the Sherburne County group. And we work with local coordinators. So that could be like the aquatic invasive point person for a county. It could be a volunteer. It could be our volunteers have done that before. So really anyone who kind of wants to take the lead on the local organization and advertising, dealing with the equipment, and doing kind of the first pass of the samples that the volunteers brought back in, can take on that local coordinator role. And through that network of all these little like satellite Starry Trek spots across the state, we end up really being able to do a lot of searching. So this is kind of what's happened over the years, and I have my little coronavirus asterisks on here as well. But we've been able to search, like last year it was 222 individual different water bodies, a couple of rivers mixed in there, 281 public accesses, and we had 206 volunteers. This was actually a nice event to have during the pandemic because it's all outdoors, it's in very small groups, and there's a very limited amount of time that you spend kind of with a group like the one that you saw in the picture before. And most of it you're spending like driving out and looking, in a very small group, which could even be just like you and a partner, or you and a family, or whatever your small bubble might be. So it's a good one to do within your bubble. Anyways, and through all of these searches, we have had we've documented 50 new reports for an aquatic invasive species, as I've kind of like spread that out across what different species people have found. Starry stonewort is, of course, like the focus of the event and what the reason that we made it. There have been 4 new discoveries of that can be attributed back to volunteers who are participating in this. Last time I did the math, I think that falls out to around 20% of all of the known populations of the state have been from this event. There's been a new zebra mussel discovery. There's been some new Eurasian water milfoil finds. The freshwater golden clam or Curbicula, those are both in Sherburne County, the 2 there. And one of those is the one that in 2020 led to that research project that Dan and I had done that we talked about at a previous one of these workshops. The mystery snails is probably the most impressive one. I'm sure there's questions. I don't know if we know that those are like new infestations versus just infestations that perhaps just have not been reported, but they had not been on like a central map or known in any sense. So at the very least, even for some of these species where maybe it's not a new population, it's actually been there for quite a while, maybe even locally they knew about it. It's still this really great way to kind of contribute back to this larger body of knowledge of invasive species distribution, and then even a couple of new purple loosestrife ones. So we do have a date set for this, and that's August 20th of this year. I think that's the 3rd Saturday of August, which is kind of where we've landed. We hold this event alongside a sister event in Wisconsin called AIS Snapshot Day. They had been doing that event focusing on rivers and streams in Wisconsin for some time. And right around the time we were thinking about trying to do something in the state that looked like that, they were really interested in expanding their program to look at lakes as well. So we co-developed this lake monitoring protocol and now hold our events on the same day each year. So the couple of links that I'm showing in the green box that says check the map, you can see where we've been. So there's little gray dots that show all of the various lakes that have been monitored over the years. You can see which year it was last monitored as part of this event and the various things that we found and where those are. And then also, if you want to find out about when registration opens for Starry Trek, that's the orange box that says get updates. If you're interested in being a local coordinator, I do have like a list of people I tend to send it to. Like if your name appears in the AIS prevention aid primary contact list, chances are you're going to get an email from me. If you have done this before, you'll probably get an email from me when we start to look for like those local coordinators on where we can hold a site. If you don't think you're on one of those lists and you are interested in coordinating locally or don't know if it's been done there before or someone else is, please reach out to me. I'd love to, we always love to see more sites go. We provide all of the stuff that you need. So we'll bring you a tote. It'll have sampling rakes in it. It'll have like boot brushes and trash bags and Ziplocs and the things that you need, clipboards to do this event. And I can send, we have like a whole list of the things that like you would need to do versus what we handle. So yeah, we hope to have more. And I have one final slide that just has contact, and I realized I didn't update this to have mine. So when I'm done, I'll put my email address into the chat, or if Tina wants to drop mine in, you can reach out to me directly. Though that email address does auto forward to me. But also has like our Facebook page, our website, our YouTube channel, which is where we have training materials for Starry Trek, but also we do like a webinar series and other whatever random things that we create end up there. So I think that's it. And I have 5 minutes, it looks like, for questions. So yeah, I'm happy to take those. I can't see chat though.
That's okay. I'll read them to you if they come through, Megan.
Thank you.
Any questions for Megan? Doug.
I wanted somebody else to ask another question, and they didn't. So
So think of it while you're asking your question. How about that?
So Megan, I really like the way that you're characterizing your volunteer value with the number of hours. It looks like based on the number of hours, you're using about a number of $30 per hour. Is that about right?
Yeah, it's pretty close. You can find it. So independent sector has a, you can, and they update it every year. So you can kind of get what the new value is. But yeah, I think it's always been like 28 to 29-ish has been the typical number.
Yeah, based on your numbers, looks like it's $29.94.
Okay, that sounds pretty close. Yeah. Yep.
We've had a lot of discussion here at Sea Grant on how to capture that value as well. And one of the other, or three other areas that we've been capturing that value is through travel time. So you can include travel time, and then you could also calculate mileage based on what the going rate for mileage is. And then the third thing is if there's any other in-kind contributions.
That's a great one. Yeah, I know we have travel time in there. We have not factored like the actual mileage in. So like if travel time is part of their volunteering, then we would encourage them to include that. And I think we have a drop-down box in our form for volunteers to enter that.
And you can use that. You can use the
probably
university.
have the mileage, but we haven't quantified that before. So that'd be really helpful and interesting too, especially with Starry Trek. There's a lot of mileage that gets put on with that because people are going to a bunch of different lakes. So yeah, that's a great thought too. Thanks, Doug.
So we've used the university mileage reimbursement.
Okay, yeah.
Well, whatever it is, 56 cents a mile.
Yeah, that's a great idea.
If people are bringing things or donating things, if they're donating food, if they're donating tables, if they're donating a display or made a display as a contribution, anything along those lines could be used as in kind.
Yeah.
Something to think about. Yep.
Thank you.
You bet.
Yeah, thanks, Doug. always good to, as a reminder, to track those kinds of things in any program that you're working on with volunteer and in-kind services, because you can really reflect how much you multiply the funds that you have with those in-kind work. Josh has a question: which mapping database are you using to document new AIS discoveries?
Those all go through EdMaps. So the EdMaps will just automatically send that report to the appropriate specialist at DNR. So it's an easy way for us to just have people put that in. And for Starry Trek in particular, the volunteers don't necessarily have to do that. They will bring anything that they think might be suspicious back to that coordinator. And then the coordinator is doing the first pass. So they have whatever ID resources. We started sending our a copy of our ID guide out as well. And if there's something that they're like really confident this is a native species, then that won't get reported. They'll mark that they got that species in, but it doesn't go into EdMaps. Anything that they either think might be an invasive species, or they just really don't know and don't feel confident on, or just want extra confirmation, they'll submit that into EdMaps. And then, you know, right away when the specialists are back to work on Monday, they have this pile of reports waiting for them. So we always give a warning so that they're ready for the pile of reports that they get after that weekend.
Yeah, they definitely appreciate that heads up, and it's a pretty slick process that we've kind of worked out between our two organizations. It's worked out pretty well. And we've also done like in-person plant and animal identification trainings, like, you know, just really brief hour or two, you know, somewhere outside in preparation for Starry Trek 2. So if those coordinators are interested in getting a little bit more familiar with identification, our specialists have hosted those kind of pre-Starry Trek events before. And Megan, let's coordinate again this year, maybe a little earlier than we did last year for those. And get them on the books, because I think those are really helpful, especially if it's been a while since you've done AIS identification.
Yeah, definitely helpful. And we always get really great feedback about those. People really appreciate you making them available. So
all right. Any other questions for Megan? All right. Excellent. Well, thank you again, Megan. And I know you probably have to run to your next meeting, so I'll put your email in the chat for everyone if they have further questions.
Perfect.
And then, so Meg, if you're there and ready to go, take it away.
Hi, everybody. I'm the other Meg affiliated with MACERC. My name is Meg Dewar. I'm the Research Outreach Specialist at MACERC. And thank you, Tina, for carving out a little time in today's agenda. I have some updates about our Stop Spiny campaign and the next steps to share with the group. So I looked at the participants, and there's a lot of familiar names. So hopefully a lot of you got an email from me the other week with all this information, but there are some new folks. So last year, MACERC ran a public education campaign about, based on our research that showed that spiny water fleas could be spread among commonly used fishing gear and in residual water. And so we developed a whole program and campaign toolkit for people to use. And so that grant wrapped up for MACERC, so we will not be able to kind of invest that amount of staff time and support with the spiny class. However, I want folks to know that the website and the campaign resources page will be up on the MACERC page more or less indefinitely, and totally encourage people to continue using those resources in their areas. And fortunately, Wildlife Forever has stepped up and most likely will be ordering the customized Swedish dishcloths. So the content and the logo will probably just be Wildlife Forever and MACERC on that. But if you are interested, I'd reach out to the Wildlife Forever folks. And when I'm done talking, I'll put in all the links into the chat so you have that. And then as far as for people that did participate in the campaign, either if you joined a webinar that we gave or ordered the Stop Spiny cloths or we provided cloths, you should have gotten the email from me that has also a survey link. And if you haven't yet, please should only take 5 to 10 minutes to do that survey. That's just going to be helpful for us if we ever do another campaign like this. So make sure we're always improving. So please do that survey. And then the last update is a new program that is sort of spinning off from this. I am working with Don Brandstrader and Valerie Brady, who are the PIs that led the spiny means of spread research. And we are developing a pilot program for 2022 that's going to be testing a citizen science approach for spiny water flea surveillance. This might be a surprise to folks, but there has not been any new spiny water flea lakes added to the infested waters list since 2016. And given that there are 66 water bodies listed and how easily they can spread, we think that they could be in more water bodies than are currently listed. And that might reflect sort of there's a lack of systematic or even targeted surveillance. And, unlike zebra mussels or maybe Eurasian water milfoil, most of the kind of the boating public is probably not going to recognize spiny water flea. It just looks like more lake goop stuck on their fishing line. So what we're going to be doing is working with any interested local manager or lake association to provide them with a protocol to effectively do spiny surveillance. Because unlike veliger tows, you don't actually, the only specialized equipment you really need is a plankton net, and they're not particularly expensive, and you don't have to send samples to a laboratory for the analysis. With a hand lens and some basic tools like a white tub or something like that, you can ID them and you can do your own plankton tows. So we will be providing some instructional webinars, an equipment list and the specifications for the plankton net, and user-friendly instructions. And we'll be, myself and the researchers will be on hand to provide support. If you're a manager in a place like Crow Wing County next to Mille Lacs or Cass County with tons of lakes and you need help prioritizing where to do your surveillance, we can help with that using some of the modeling data we have. And what participants will have to contribute is their own gear. We don't have a big grant to provide sampling equipment, but we think it's pretty affordable. The whole shebang should cost less than $200 or thereabouts. And then you would need a boat. So another important thing with this will be guidance on time of day and time of year to maximize your detection probability. And then of course, we'll have a system for reporting both positive and negative detections. And we'll be collaborating with the DNR to make sure that we're efficiently communicating that information, providing good enough voucher, either specimens or photos to validate that. And then, the only other contribution we'd like from participants is feedback on our materials and methods as we develop it. This is a pilot approach, and so collaboratively kind of talking through this and working out the kinks is really important. And I will put my email in the contact. I'm right now just building up a list of folks to get updates. We have a draft of the protocol right now that's in review, and we'll be sharing that shortly. And then I'll send a link to a little flyer with kind of an overview of everything I mentioned. So yeah, sampling we're going to be recommending for August and September, most likely. So we got a little time. Okay, that's all I had. Thanks, Tina.
Yeah, awesome. Happy to have people know about this project since it's very relevant to the work that everyone here is doing. So thanks for that information. And if you have questions for Meg, you can send her an email, or feel free to send me an email and I can get it to Meg. Either way, we'll get it out there. And yeah. So I think now we will take an 8-minute break to keep us on time for our last two presenters. So we'll come back here at 2:20 p.m. So thanks, everyone. I encourage everyone to stand up and move away from your screens for the next 8 minutes. See you soon. Hi, Chelsea.
Hi, Tina.
It's 2:20, so I assume everyone is exactly right back in their seat just in time. No.
You can see my screen now, right?
Yep, I can see just
Cool.
the first slide there. So if everyone, if you're here, you want to just turn your camera on real quick or give a little thumbs up or raise hand just to let us know you're back here. Excellent. Cool. I saw a ton of hands up and a ton of thumbs up. So I think we're ready to go, Chelsea.
Awesome. Well, yeah, thanks, Tina, and thanks everyone for being here to think a little bit about trade pathway invasive species prevention. So I'm Chelsea Blanke. If we don't know each other, I've been working for the DNR's invasive species program on trade pathways for invasive species in this 3-year federally funded position that is actually expiring mid-June. We know that global trade drives invasive species introductions to Minnesota and the United States. So we're at this another turning point here for trade pathway invasive species prevention in Minnesota. We've learned a lot about risks associated with trade of live organisms in the past few years, building on past work. And there's still a lot more to learn, but I think we're at a point where we do know enough to really start to ramp up prevention programming for target audiences in these trades, like sellers and buyers or hobbyists. So I'm excited to be with you here today. Hope we can think about how to keep up the momentum toward preventing invasive species introductions through these pathways. And I think invasive species, local pros like yourselves can play a big role in this going forward. So I'll clarify that the trades that we've been focusing on are those that rely on the movement and possession of live organisms. I'm going to share findings from some of the DNR's recent projects, but the trades that we've been focusing on are the pet, bait, food, horticulture, and classroom and laboratory biological supply trades. Major projects that we've been working on included workshops and surveys of live organism businesses and hobbyists, an in-store assessment of the availability of invasive species at pet stores and seafood markets, and development of online and print educational materials. So just a little bit of background about kind of the risks associated with these pathways based on the agency and scientific literature that's out there, conversations with experts around the country working in this area, some searching for invasive species online. We know that about 42 or 2/3 of Minnesota prohibited invasive species, the ones that are illegal to possess, have been documented in these trades. 15 or 3/4 of Minnesota regulated invasive species have been documented in these trades. As a reminder, those regulated species can be thought of as somewhat akin to a watch list, but they are legal to sell in Minnesota. And around 15 additional risky species, which are unlisted, but risk assessments have suggested likely harmful impacts to the Great Lakes region, have also been documented in these trades. Some additional issues are that release from these trades is not uncommon. There are some great outreach campaigns out there, but they're not always recognized by individuals in the industries or their customers. Species are often mislabeled or unlabeled. We don't often know what these supply chains look like. You can buy lots of invasive species online, and authorities for inspection and enforcement are somewhat limited. So I'll dig into details on a few of our projects here. We developed some online and print resources on trade pathways generally, some that are also specific for sellers in each individual trade as well as responsible buyers. So if you haven't seen these, I recommend checking them out. You can just search for Trade Pathways Minnesota DNR. And hopefully, if you're interested in working on trade pathway invasive species prevention, these materials provide some some messaging that you might be able to build off of. we've actually put a substantial amount of effort into educating folks about the issue of Louisiana or red swamp crayfish in crayfish boils. This prohibited invasive species is commonly used in boils, and many people were not aware of the laws, have been importing them live into the state. So these crayfish are causing serious problems in Michigan and Wisconsin and would likely be impossible to remove if they were to become established in a Minnesota lake. In the past couple of years, we've notified distributors in other states and local restaurants and markets about this issue. We've also done multiple news and social media communications about the issue more broadly. So if you see an event or an advertisement for an event where folks are planning to use live Louisiana crayfish, please do let us know about that. That's not something that we permit folks to do. It presents an unreasonable risk to our natural resources. And we can provide them, and you can help provide them with alternatives like using native Minnesota crayfish, or they can get permits to import frozen or boiled Louisiana crayfish. We actually had about 180 inquiries about this last year and issued over 100 permits for use of frozen or boiled Louisiana crayfish last year. So this is a fairly common activity. We have also done a direct mailing to more than 3,500 businesses and invited them to complete an invasive species survey. Just some gains from doing that mailing itself were just helping us to educate sellers about invasive species regulations, let them know of that new website content, and inviting them to join email lists so we can have a better way to stay in contact with them in the future. In that survey, generally, we saw that most sellers are concerned about and take responsibility regarding invasive species. We did have a subset of sellers within each trade that did not think this was a concern or something they needed to be involved in. So we do need to be conscious of these differences in opinion, try to meet folks where they're at. And we learned that the majority of sellers are not providing educational materials to their customers, but most businesses would be willing to distribute invasive species educational materials to their customers in the future. We also conducted a retail assessment last year. This is the project where we hired Fortin Consulting to visit Minnesota aquarium retailers and seafood markets and examine their stocks for invasive species. They also spoke with and educated the store staff. So I'm not going to go into the findings from this too much because Hennepin County pet stores were visited as part of a separate project funded by Hennepin County's AIS program, and the finding from the Hennepin County project really mirror the findings from other parts of the state, just at different scales. So I will say, though, that we found prohibited invasive crayfish at multiple pet stores. There was evidence of other prohibited invasive species being sold at pet stores and seafood markets. And all the pet stores that were visited did carry some invasive species. Tony Bra with Hennepin County has been a real leader in this type of work for several years, so I will let him dig deeper into this in the next talk. And yeah, I mentioned that we conducted a hobbyist survey, aquarium and water garden hobbyist survey this past summer. And this was actually led by Tina, so she could give this part of the talk as well as I could, but we worked with Ken Donnelly on our community-based social marketing contract partner on this. A total of 479 respondents completed the survey. And the survey was distributed through the DNR's AIS communication channels and hobbyist groups like the Minnesota Aquarium Society and Water Garden Society. So we do want to acknowledge that these folks, might be disproportionately aware of invasive species and concerned about them. So we also worked with U of M extension programs and pet and garden stores in the Twin Cities area to get the word out. So I will jump into some of the results from this hobbyist survey. We asked if the respondents knew what invasive species are, that aquarium and pond species can harbor hitchhikers, that some invasive species in the trades are invasive, or some species in the trades are invasive, about regulations prohibiting release, possession, and sale. And we saw pretty high levels of awareness, as you can see here across the board for these questions. We also asked about attitudes. More than 90% of hobbyists agreed that preventing invasive species spread is the right thing to do, and that aquarium and pond species should never be released into the wild. Just over 80% indicated they were interested in learning more about how they can help prevent spread. So one of the recommendations from that hobbyist survey report was to highlight the high levels of awareness and positive attitudes as social norms in our educational materials. Behavioral psychology research suggests that establishing prevention behaviors as social norms can encourage others to practice those behaviors too. So despite the generally high understanding of the basics of invasive species regulations, only about a third of respondents knew which species are illegal to possess and sell in Minnesota. And many reported that they felt they did not have or don't know if they have the information they needed to avoid the purchase of invasive species. So this suggests we have a lot of work to do to help educate hobbyists about which species are prohibited and present risk of invasion. A couple of important hobbyist practices that we asked about were what species they have had in their aquariums and ponds. For aquarists, around 20% have had crayfish, around 18% have had turtles, and around 70% have had snails. For water gardeners, around 11% have had crayfish, though we suspect that many of these in ponds are native crayfish that moved in naturally. Disposal in the trash and composting are the most common methods for disposal for plants that hobbyists no longer want or can no longer care for, followed by sharing or trading with other hobbyists. More than half of hobbyists check for hitchhikers and half quarantine new plants and animals before putting them into their aquariums or ponds. So this shows that hitchhikers are very common to the point where it's common practice for hobbyists to check for them. And this issue with hitchhikers is something that has been shown a lot in past research as well. And lastly, 21% of water gardeners reported having ponds near other water bodies. Thankfully, though, very few reported flooding where they have their ponds. We asked aquarium and pond owners what they would do with animals they were no longer able or willing to keep. So this is a sensitive topic. However, most pond and aquarium owners said that they would be willing to humanely euthanize animals and would be more likely to do this than release them into the environment. We also asked if hobbyists would go to great lengths to find an alternative to euthanizing them or releasing them into the environment. And I wanted to ask this question because sometimes these are the only options that we have for folks when it's not easy to share or trade with others. So you'll see there was some greater uncertainty and disagreement in response to this question. Around 15% disagreed. So to me, that's not an insignificant risk of release if that is, how we're interpreting those responses there. Some respondents suggested that there should be more rehoming options, and they would prefer to rehome than euthanize. And interestingly, some said they became willing to euthanize aquatic species after seeing news stories about goldfish released into natural areas. Aquarium and pond hobbyists are willing to make commitments to preventing the spread of invasive species. Behavioral psychology research finds that people who commit to taking action are more likely to follow through on a behavior than if they had not committed to it. So survey respondents were shown one of the 2 example pledge forms here and asked if they would be willing to complete the form. And around 70% said they'd be willing to complete the simple form on the left, and around 78% they said they would be willing to complete the more detailed form on the right. So customer commitments are something we could experiment with in developing educational materials for customers. We asked how they would prefer to receive invasive species information. The most preferred places to receive information were through internet searches, at the point of sale, through brochures and social media. And interestingly, around 30% would like to receive information at fishing piers and boat launches. So there could be an opportunity to utilize existing outreach efforts there that you are involved in. And full reports from that hobbyist survey and our retail assessment are available on our website if you're interested in digging more into all of this. I just want to touch briefly on online sales and issues that extend beyond Minnesota's borders. I mentioned, I want to mention this so that all of you are aware of ongoing efforts if you're ever asked about this by a business owner or a hobbyist. So the Great Lakes Detector of Invasive Aquatics and Trade, or the Gladiator Project, was aimed at reducing the availability of invasive species online. It was led by the Great Lakes Commission, and it used an automated web crawler software to search the internet for sellers of a list of high priority invasive species. So this project provided a reasonable means for identifying these sellers, which I feel is extremely critical for invasive species prevention. So we're hoping that this project is continued in some form. There's also a need to work with major online marketplaces like Amazon and Etsy, eBay, and there may be a need for increased regulation and enforcement. So there are discussions happening nationally about the online sales issue. And many of you are likely aware that zebra mussels were found contaminating aquarium mossballs last spring. And I wanted to just acknowledge this response that occurred last year as an example of partnerships across agencies and scales that worked really pretty well. So now what? To wrap up, I'll just say I think there's so much more we still have to learn with regard to these pathways and how to best address them for invasive species prevention. A lot of work has been done, but now we're, like I said, we're at this turning point, and we can really start to take on, our whole invasive species prevention community can really begin to take leadership in piloting new programming in these pathways. So if you have an interest in being a part of this, here's some initiatives, just some ideas of things that you could be a part of. Most of these were major recommended next steps from the projects that we recently have completed. So we need to continue to build a database of sellers. We have a start on this, but this was actually a pretty substantial undertaking to begin with. We don't have a great way to track some of these businesses. They're not necessarily licensed, depending what trade we're talking about and things like that. So getting to know the stores in your area and starting to build those relationships, working with us so that we all kind of can stay connected is something that is a need right now. As you're building those relationships, even if you're just popping in once a year and having a simple conversation about invasive species, could be a really good step at this stage. You could hand out print materials that we have on our website, things like that. I know some of you are doing this kind of thing already or planning on doing this already, which is really awesome. There's an opportunity to partner with retailers to jointly deliver these programs. Could be just handing out information or like working with the seller to provide information to customers, potentially experimenting with those customer commitments. So there's lots of things that ideas there and things that we could talk about. Reporting prohibited species found in stores or online. The DNR's authority for some of these pathways relies on reports from the public. So if you happen to see something and you're visiting a store, please let us know and we can help with that. Another opportunity is fostering rehoming and take-back programs. Minnesota Sea Grant has partnered with the Minnesota Aquarium Society to host surrender events where folks can bring in animals that they can no longer care for, give them kind of an out from releasing them into the wild. So, perhaps this is something you could think about organizing in your area 1 or 2 times a year. I believe Sea Grant has guidance materials on how to put something like that together. And last couple ones are connecting with teachers on the issue of release of classroom organisms. We've done some work with biological suppliers, but this is something we have not had a chance to get into much yet. So there's definitely some opportunity here. And county fair outreach and reporting. Some of you may remember that we did education to fair organizers a couple of years ago, and we did that outreach again this year. So we'll be doing another kind of survey to see if there's been any change in offering pets as prizes at these fairs, like goldfish and red-eared sliders. So there's lots of things that can be done, and a lot of it might not even need a ton of time or effort. If you're thinking about this, but you're not quite sure where to start, please, give me or Tina a call. We'd love to work with you. And yeah, I know I'm out of time here, but just want to acknowledge the many individuals who supported this work. And thanks you all for being here.
Thanks, Chelsea. We can spare a couple minutes if people have some urgent questions they'd like to ask. But yeah, like I said, like Chelsea said, reach out to either of us about any work you'd like to do about trade pathways for aquatic invasive species prevention. We'd love to talk with you more about it and have lots of resources we could probably share with you or help create. So. And there's Doug's contact information on the side there for rehoming and surrender events. Awesome. Thank you, Chelsea. Tony, you're up.
Thank you. Am I sharing?
You are sharing the screen with everyone's video.
Nice. I'm going to close my video. How about now? Let's see here. Oh, I'm going to just do it again. Anyway, definitely not one of my fortes here. How we doing now? We sharing, right?
Yep, it is now on just your first screen there. So thanks for presenting, I think, at literally all of these workshops, by the way, Tony. So
do I get a check in the mail or anything?
Yeah.
No, I didn't present one because I was in the British Virgin Islands sailing. So
okay.
This project is something that we've done because actually it was really nice. Address all pathways was one of the guidance that we got originally way back in 2015 when we started our program here at Hennepin County. And you can kind of see this is why we do this one. It hits quite a few of our strategic goals that the county has. So yeah, the story brings me all the way back to 2015. And we initiated kind of a search for pathways other than watercrafts, and that brought us right to pet stores and nurseries throughout Hennepin County. In that year, we did it incognito, so they didn't announce that they were in there. They basically just walked around and looked to see what was for sale. And 88% of the pet stores carried at least a regulated species, 94% a Minnesota regulated aquatic plant. 20 of our stores had goldfish, 6 had koi, and we had 2 that were kind of early detection targets at the time, water lettuce and yellow floating heart. And then we had 1 store with a prohibited species labeled, but then we bought the plant and we looked at it and it wasn't that species, it was something else. But that does indicate that, they would have sold a prohibited species if they actually had the right plant labeled. So that was 2015. In 2016, then we kind of announced our presence and we gave them a little more educational information, specifically from what Doug does there at Habitabutude. 2017, we identified stores both in Hennepin and Dakota counties selling Oriental weatherfish, kind of I think it's a bottom feeder, but it's a prohibited species. It was also sometimes labeled as, I'm not going to say this right, gold dojo loach, I believe. And then there was 2 other stores that had it, but it was out of stock. I never did find out exactly what happened because we did contact the DNR and talk to these stores a little bit. So I, a year later, called them up and I said, hey, this is what I'd like to buy this from you. And they didn't know who I was. And he goes, hold on for a second. He came back and he goes, no, we can't sell that anymore. And I was like, all right, it worked. So we accomplished something there. So in 2018, we actually did some interviews, kind of surveys, I guess I would call them, with 26 garden center stores. And we asked them kind of a series of questions. I'm going to go through these pretty quick, and you can do a lot of the reading on your own here. But we asked them, how do you dispose of it? And then we asked the pet stores, how do you dispose of it? And you can see the answers there. One of them, though, said we throw it in the backwoods. One store said that. We asked them, how do you dispose of your water? One store did say asphalt parking lot. With the pet stores, how do you dispose of your water? And there were kind of the answers. And we had one store that in the parking lot and one down in the storm drain. We asked, do you have a store policy for disposal of unwanted plants, animals? And, you know, the majority said no, and some said yes. And then 6 of the aquarium stores said yes, and the rest said no. Then we asked, does your store accept unwanted plants? And most of them on the garden center were returns only. And as far as the pet stores, there was a lot of different kind of answers here. One store said they would accept anything, which was nice. Next question was, can you explain the difference between a prohibited and regulated species? And 14 of the 26 garden centers basically said no. And some of them here are right and some of them are wrong on their answers, but at least they didn't say completely no. And then with the pet stores, we had 5 stores that said yes and 12 that could not explain the difference between the two of them. Garden center, or in the garden centers, we asked, would you be willing to remove prohibited species and regulated species from your store? Kind of a very interesting question. And most of them said it's a corporate decision. And I think we got the same kind of answer with the other ones. Most of them are going to be a corporate decision. And a few said yes, sure, no problem. Probably doesn't matter, owner's decision, that type of stuff. Are you familiar with habitatitude? Doug? We had 2 out of 26 that knew about habitatitude. And then we, you know, we asked them would they be willing to distribute the materials? If they have any materials. And 23 out of 26 said no. And then 2 of them said verbally discuss, and I can't tell you what the one is that's not in there. Interesting. And then same thing with the aquarium stores. Most of them were not familiar, but a few of them are. That's exciting. And 17 of the 17 stores really didn't have anything that they hand out. Most of them just do it verbally. So in our county, this is kind of a map of what it looks like. These are the pet stores that I have throughout Hennepin County. So we got 20 of them. And here's our garden centers with 14. And so 2021 now, we did another sweep through all of the garden centers and pet stores last year. And we did find 1 prohibited, 1 proposed prohibited, 1 early detection, and 7 regulated AIS on this search. So 95% of the pet stores sold goldfish, and 17 of them, so 85%, sold koi. And here's kind of some of our findings that we had. Red ear sliders, I want to say that's a turtle, if I'm remembering right. And you can see we've had some that were doing, you know, water lettuce, and yellow floating heart was found in 1 garden center. And then hitchhikers that Chelsea talked about. Chelsea, you're my new favorite. She hit it right on the head here that it's definitely a topic to talk about because there are things in this plant. A lot of it was little tiny snails. So the other thing we noticed on Nextdoor, and I really don't know what Nextdoor is other than a way for neighbors to talk to neighbors, is we did find 1 person posted that they were willing to sell frog bit through this app. And then we found red swamp crayfish for sale. By the way, I think it was like $50 a crayfish. So they're not cheap. And they found them labeled in 2 other stores, but they didn't have any present at the time. And so that was kind of a big find. I think Chelsea found kind of a few more out of Hennepin County. And we did work with the distributor and the stores, and I do believe that they've stopped selling them. So one note that's coming soon here in 2022, we had a grant application come in, and we've agreed to fund it, is we're going to do an AIS online course for pet stores and garden centers. And it's either going to be like a PowerPoint or a video that their staff can go through. It might have a test. It most likely will have a pledge. There might be a certificate. It's still being exactly kind of flushed out how we're going to handle it, but it's going to be voluntary. It's not going to be like a blaming thing. It's going to be more like education and awareness and 100% positive to try to get a little more working relationship with these companies in our county. So that's what's coming this year. And there's money. So that's all I kind of had for you. I am completely open to Chelsea getting back online too and answering any of the questions that come that I won't know the answers to. And Doug, you can't go first.
His hand just popped up as you said that. Thank you for sharing. Very excited to see what you've done and where it's going as well.
Okay. Doug,
Any questions for Tony?
Go ahead, Doug. What you got?
That is way overinflated cost for a red swamp crayfish.
Really?
Way over.
You don't live in the metro.
You can buy them for $6 a pound off the internet live. So they're making a heck of a profit off of those red swamp crayfish until they were confiscated.
Interesting that they were finding the red swamps there. The colorations were nowhere near what the typical red swamp really looked like in the ones that we've found. So
that was very different. Yep.
Yeah.
Very different than any that I've ever seen too.
Yeah, let me talk about that a little bit. So we actually purchased these because of that. And we shipped them out to Michigan, and I don't know who, but a specialist out in Michigan, and they confirmed that that was what they were. So we did actually purchase, I want to say 3 of them, and sent them out there because all 3 were different. And they confirmed the correct species. So.
Yeah, Tony, in response to your exposure question, so when the campaign was launched back in 2004, there was a lot of interest by Petco and PetSmart, and they integrated a lot of different outreach materials into those stores. But because of lack of funding for the effort, it really fell off by about 2007, 2008. So those consumers and pet stores that you were contacting haven't really been exposed, especially in the Twin Cities area, unless they attended events in the Twin Cities where we've been promoting Habitattitude. But most of the promotion has been up here in the Duluth area where we've hit 50, 60 events per year with Habitattitude, and also had good cooperation from the stores in our area.
Yeah, and even though it wasn't being run that much in 2016, we did bring the Habitattitude information around to these stores and handed it out. I think we might have made it branded Hennepin County, but it was basically that information, and it did include the word Habitattitude and that information from the program.
Sure, it's like anything else. If there's not the constant exposure, people will forget, and it drops out of their memory.
So I actually would like to challenge every county to do this, because a lot of your counties don't have a lot of stores, so it wouldn't be that big of a project. But Ramsey probably has a lot. I would think, as you get in the metro here, that we have quite a few stores. But you get outstate, there's probably only 1, 2, 3, maybe one in this big city and one in that big city. So we could do this, and yes, it's never going to be 100% foolproof because we got the internet that you can buy stuff on, but at least it's the start. And even if somebody buys something on the internet, they often would go into these stores for advice or some product or whatnot, and then hopefully they would get the idea there, like, okay, when you're done with it, don't put it in the storm drain, don't put it in the wetland, don't put it in the lake, and at least that would hopefully slow things down a little bit.
Exactly. Yeah, kind of attacking things from all sorts, all sides there. If
you're wondering on budget, I would say every year these projects were anywhere from 15 to 22,000, depending on how much we did.
Yeah, thanks. That's helpful, Tony. Any
Tony,
other questions?
I agree with you.
Oh.
if you went to if we went county-wide and got these programs set up, but also the Habitattitude surrenders have also been very successful. And I've either led or been a part of one with the Minnesota Aquarium Society in 17 events since 2017, and we've rehomed over 1,200 animals just based on those events. And they're very easy to set up. The work is, the labor is up front, so it takes a little bit of time on the front end, but the actual implementation costs, if you can get a place to be the host for the event, doesn't cost a whole lot of money.
Exactly. Thank you, Doug. There is one question about reaching out. Have you reached out to Walmart?
We went to all the big stores, but I don't remember our Walmart selling plants or animals. Is that typical, Michelle?
And Chelsea, do you have any comment on that?
Yeah, I was just gonna chat, but I think that they actually stopped selling aquarium fish in 2019.
Okay.
So if you haven't, if you've seen them since, we'd be interested to know that. But that's what I heard. They had a lot of like animal welfare concerns. So I think they stopped in response to that.
Okay, it's good. It's good to know. I just wasn't, I hardly ever go into my Walmart, especially due to a pandemic. But the last time I was in there, I just knew that they had fish, so I didn't know if there was other things that they had. But I definitely will, next time I go into my Walmart, I will take a walk through. It'll be interesting to know what they are selling.
Some of them did stop selling, but they're still in the United States, they're the third largest retailer for fish.
All right. Well, definitely something to keep an eye out for then.
And what they generally sell is the most common fish, so that they can move them fast.
All right. Yeah, Chelsea.
I just wanted to add to, I guess I really like Tony's challenge to the counties, and Tina and I, when we did the hobbyist survey and visited a lot of the metro area stores, it was just a really pleasant experience. They were, most of the retailers we talked to were just, happy to have the information. And I think, doing more of a study might have a little bit more cost to it, but just, a lot of most of these pet stores now, we went to, between the two projects, Hennepin County and the DNR one, all of these pet stores in the state that we know of have been visited. So they've kind of been primed on this. So, even just like I said, just at this point, a couple of little reminder visits might be, just the ticket for, where things are at right now.
Yeah, and building those relationships with local business owners, and if they find something weird as a hitchhiker or have a question about what they can or can't sell, they have a couple contacts now to reach out to. And that's the key right there, is to get people just talking about this issue in general. And then the DNR is hopefully going to be creating more publications and more resources to become more widely distributed on these trade issues specifically. So
I do have one question. We did not do the food markets. You did. Anything? I didn't pick up on it. Any findings there?
Yeah, so we did have several that were advertising red swamp crayfish, either on their websites, or they had a tank labeled for crawfish, but they weren't observed by Horton Consulting in person. So we did do some outreach about this last year, so it could have been they were picking up on that. I also heard last year there was a lot of serious flooding in the south, and so that could have affected the Louisiana crawfish are kind of a very seasonal commodity. So I think they might have been just hard to find in general last year. So just because they weren't observed, I don't think that necessarily means there's no risk there.
Yep. And all of the information from that surveillance report, I assume, Tony, you have your report online, or maybe it will be online. And then we also have our DNR report online, or you can share it with me and I can share it with people that are interested, or they can just contact you. And there's a list of all the different species. I think there was like 500 species of fish being sold at these stores that we found. And so Emily, I see your question in the chat there about alternative names, so we can talk about that. But it is 3 o'clock, so I am going to wrap us up for now, but if you want to stick around and ask more questions about organisms and trade, Chelsea and I could stay here all day and talk about it. So I did want to just put in the chat here that I did record the very first meeting we had, the first workshop we had on behavior change in early February, and I finally got that recording all ready to go and share. So that's in the chat right now for those that might have missed that one. This one has also been recorded, so we'll get that shared soon as well. And just wanted to thank everyone for attending and participating, and a special thank you to all of our presenters on all of this awesome information. I know it's a lot to take in, and all of us are always willing to talk more about the stuff we're into, so feel free to reach out and connect with us. So thanks, everyone. And if you want to stay around for an informal discussion about organisms and trade, you're welcome to. Oh, and there's your link. Thanks, Tony.
It is online.
Thanks, everybody. Thank you, Tina. This is great.
Thanks, Meg.
Chelsea, we'll be calling you before you leave to help us with our
Cool
project.
Call me soon.